This week we are going to be learning about DNA isolation from cell culture and the different techniques we can do with it in order to analyze the extracted nucleic acid. So why would we want to extract nucleic acids? Well, the biggest reason to extract nucleic acid is to identify whatever it is that we're extracting the DNA from. That's one of the main goals that we usually have. It could be used to detect and identify bacteria or viruses in the environment or in a sample from a patient that we are trying to diagnose for a particular disease. So that would require DNA or RNA extraction as one of the early steps. In addition, we can also use it to diagnose diseases or genetic disorders where mutations have occurred in the nucleic acid as well. In order to do that appropriately, we would need to know the sequence of the genes and which genes it is that are involved in that process. Other reasons we want to extract nucleic acids is to study a disease process simply by actually sequencing the DNA or the genes associated, the exact genes or regions associated with a disease, and then studying the mutations or changes that are seen under various environmental conditions or under a normal versus a disease state. What we can use to extract DNA is actually pretty much anything. Because of the advances in DNA technology or nucleic acid extraction technology, we can pretty much extract DNA and get a whole blueprint of a person from, or a fingerprint of an individual simply from a single cell, as long as we can find one. And so you can get these cells from biological specimens, obviously, from actual cells or tissues. You can get it from blood, from semen, from saliva, urine, hair, anything, teeth, bone. You can even get it from non-biological material that have come in contact with the individual. So that includes cigarette butts, envelopes, stamps, anything that could have been touched or has cells left over from that individual. This is why it is used as biological evidence for cases as well, right? In forensics, there's a big, huge need for forensic biology and people who can do exactly that. Extract DNA and make fingerprints of individuals for forensics needs. So how do we do this exactly? Well, at the end of the day, the procedure is pretty simple. You start up by opening the cells, obviously, because you want to get inside the nucleus where the DNA is. So you break open the outer cell membrane and the nuclear membrane. You then use agents to dissolve those membranes so that you can remove them from the system and remove that contamination. We also want to extract the DNA in as pure a form as possible, which means that we want to remove any other stuff that's there. Majority of that stuff is going to be protein. So we have to separate or remove this protein fraction either by destroying it or by, removing it in some way, shape, or form. The reason for that is that, well, obviously the protein is going to contaminate your DNA, but it also contains enzymes that can chew up the DNA called nucleases. And so by removing the protein fraction, either by just separating it and removing it or by denaturing it or destroying it some other way, you are protecting the DNA that you're extracting so that it does not get chewed up by the nucleases and get destroyed. And then finally, you want to precipitate your nucleic acid, whether it is DNA or RNA, so that you can concentrate it in the amount that you need for your downstream activities that you need to perform. We do that by adding alcohol to our remaining mixture after we've removed insoluble matter and the protein fraction. The reason for that is that alcohol lowers the solubility of DNA inside your solution and precipitates it out. It also removes alcohol-soluble salts from your system so that it's more pure DNA that you get as a final byproduct. In our lab setting, we use a kit, a thermoscientific genomic DNA purification kit, that is based on chloroform ethanol precipitation method. In this case, after we lyse the cells, so break open the cells, we heat our fraction to denature as many of the proteins as we can and separate them from that surrounding, from other proteins and from DNA. Then we add chloroform to the mixture to emulsify the mixture. And what ends up happening in that case is that you get your protein fraction, your organic layer and an aqueous layer. And your organic layer is going to contain a lot of the proteins in them. The aqueous layer is going to contain your DNA. And then you can precipitate your DNA and then get redissolve it, resuspend it in solution in whatever you need based on what it is that you're going to do with it. Now, when you precipitate the nucleic acids, you can technically get both DNA and RNA out from the same cell. To make sure that you're only getting the DNA fraction or the RNA fraction, we can selectively treat our precipitated product with either DNAs or RNAs so that we just get DNA or RNA out, precipitated out as needed. However, in many cases, RNA is extremely fragile, so it does get degraded simply through the process unless it's extracted in a very careful manner using agents that protect it from degradation. Now, in the phase separation, like we talked about, we are using chloroform to emulsify our lysate and to separate it into the organic and the aqueous layer. What you end up with is an organic phase which contains your proteins, your interphase which will contain some insoluble matter like the lipid membranes that have not been dissolved, and then you have an aqueous phase which contains your nucleic acid. You then take this aqueous phase carefully by pipetting it into a separate tube and add alcohol to the mixture to precipitate it. Now, one thing that I have not talked about in detail is the actual lysis of the cells. To lyse the cells, you basically want to permeate the cell membrane. If you remember, cell membranes are lipid bilayers, so they're just a lot of fat in them. Well, just like Dawn dissolves fat and grease from our dishes, we can use detergents to dissolve the membranes of the cells as well. Similarly to how with COVID-19 or any infection as well, we are asked to wash our hands with soap to destroy the integrity of that membrane. So we do exactly the same thing. The lysis buffer contains detergent as one of the main components. And then it also contains buffers to protect the rest of the stuff as well, certain salt concentration and things like that. So that is something important for you to know. In this case, that detergent is a main component of the lysis buffer that we use in order to destroy the cell membrane and make it permeable so that we can get the contents of the cell out. Okay, so this is pretty simple. Basically 3 steps. You lyse the cells, you separate the different phases, and then you precipitate your DNA. You can do that with household materials, obviously, as you saw in the protocol that I loaded for you to try out. However, that's not going to give you a super pure DNA, which is what you need for any downstream activity that you would need to do in a molecular and cellular biology lab. To maintain the quality of your DNA, we have to use the biology, the lab grade, research grade materials and specific steps to remove contamination as much as possible and to remove the proteins and to remove the insoluble matter. So what you end up with is pure product. The more pure the product is, the easier your analysis would be. We would talk more about the analysis in the next video when we talk about quantitation and quality analysis.